we left off last time talking about the difference between Shannon information and functional information. So I want to return to the coin example here. And I really should get a better example than this. Shannon information, remember, is any ordered system, right? If you've got something ordered in a certain way that excludes other options, you've got information at that point. But Shannon information is just that. It's like the basic level of what information has to be. It just has to be ordered in some way, right? An additional level of information would be if it actually is functional, if it performs a function or a purpose or communicates something instructive, right? So here is some randomly tossed coins. And if heads is, if we translate the coin into digital information, zeros and ones, heads is 1, tails is 0. So this would be 1, 0, 1, 0, 1, 0, 0. Sorry for the resolution there, but that's a tails, so it's a 0. That, if you just did that and just generated that random coin toss, you've got some Shannon information, right? Because for every position, you've chosen 1 and excluded 0, so we have information, okay? That just in itself is just purely Shannon information, right? We can represent that by probabilities of how often that would come up. But what makes this now functional information is if I told you something about what it means, okay? It becomes functional information if I tell you that that 7-digit ones and 0 pattern is actually the ASC2 code for T. Okay, that is the digital code in ASCII. That code's for T, right? 101, 0100 in digital code means the letter T, okay? So these 7-digit packages is what codes for alphabetical language in the ASCII code, right? This has now become functional information. Okay, it's no longer Shannon information because we now have a code and it's turning into something else, right? The same with the dice example. You could randomly throw dice and you could get 4, 2, 6, 6, 1, 2. That's Shannon information, at least. It's functional because it also codes for the Minneapolis area code, okay? So do you see, we're kind of layering levels of information here, okay? Information, like I said, is determined by protocols, by rules, right? We've established a rule that says this pattern of 1s and 0s means T, right? It's not because the coins had to flip that way by any law, right? You got equal chance of doing either one if you're just randomly tossing coins, right? So there's no chemistry, there's no physics, there's no anything that determines that that's the order they have to be in. The thing that makes this functional information is that we've agreed upon it. We've created a code, all right? So the question I pose is, what is the origin of biological information? Because just like 0s and 1s are coding for T, the letter T, also the codons, triplets of nucleotides in genetic information, codes for protein. That's functional information. So where does functional information come about in biological systems? Here's a quote from Carl Sagan. Carl Sagan was a devout atheist, naturalist, no friend of Christianity. But he's talked about the complexity and the information content in cells. So here's a quote from him. He wrote the Encyclopedia Britannica entry on life. So in the Encyclopedia Britannica, if you open it up to life, Carl Sagan wrote the article. And in that he said, the information content of a simple cell has been estimated to be around 1,012 bits, comparable to about 100 million pages of the Encyclopedia Britannica. So that's actually, so 100 million pages of the Encyclopedia Britannica is equivalent to the amount of information that's in the simplest cell that we know. That's a lot of information. So it begs the question, where does that information come from? As an example, here's Mycoplasma genitalium. This is a tiny little bacteria that lives on the skin cells of mammals, organisms, mainly in warm regions like the genitals, so thus genitalium. It's one of the simplest organisms we know. It's got the smallest genome. That's what makes it significant. It's just random that it happens to be on your genitals. 562,000 base pairs in its genome. It's like just a little over 500,000 base pairs, which is a really small genome. It only codes for 482 proteins. But that's still a lot of information, right? It's just a small genome, small number of proteins necessary. But I'm going to talk about here how much information really is contained in every single one of those proteins. Okay, so I'm just giving you an overview. We're going to talk about proteins in cells. Now, in order to actually get a functional cell, you've got to have lots more than just DNA and proteins. You need properly folded, intact proteins that are necessary for replicating the genome, for translating, transcribing. You need tRNAs, ribosomal RNAs, amino acid transferases, all have to be folded in their proper function. And you need nucleotides and ATP. So you can't just put DNA and protein, or DNA in a membrane and expect that to be an alive cell, right? You've got lots of other things. But I want to narrow in now and talk about how do you actually get biological information. I am a theist, and I believe that God believe that he created cells. For a naturalist, though, you have excluded that. So either you're a naturalist and you don't believe that God exists, or you're at least a functional naturalist and say, well, even if God exists, he didn't do the creation of the information. Either way, you're left with only two options, really, for where information comes from. The first option is it just arose by chance. That the functional information in DNA, the functional information in proteins, only arose by chance. So I want to think about what that means for a single protein. Let's think about a theoretical 150 amino acid long protein. Okay? So that's a pretty short protein. Actually, most proteins in your body are thousands of amino acids long, but we'll think about an easy one. Okay? To get that protein sequenced by chance, you have to have all 150 amino acids be in one form. If we're just talking about pure chemistry, just amino acids forming and bumping into each other to form a protein, amino acids can occur in either, you've all taken organic chemistry, it can either be the D or the L isomer, right? And chemically, those act identical, right? So you have to get all 150 of them in the L isomer form. The chance of this happening just purely by chance is 1 in 10 to the 45 power. Okay, that's just getting all the amino acids there in the proper left-handed form. Because in cells, we only use amino acids in the left hand. We don't use the other isomer. Now, to get all of them to form, so they're just bumping together, forming bonds, right? You don't have amino acids just randomly polymerized together. They're all polymerized together through the N to C terminus through a peptide bond. Okay, so that's one of several possible linkages you could make between two amino acids, right? So they all have to be in that peptide form. Again, we're talking, all of them being in the proper form. Another 1 in 10 to the 45. Okay, this is a lot. A very, very rare chance, right? 10 to the 45 is a huge number. We're not done yet, though. That's just to get all the right amino acids in the L isomer form and forming peptide bonds. If you actually get that peptide to fold in a functional way, right? If you have a 150 amino acid polymer, that can fold in multiple different ways. And you know that the protein's three-dimensional structure is what determines its function, right? If you take a protein and you boil it, and you just denature the whole thing into a floppy noodle, it no longer does any of the function it used to do, right? It has to be formed in its proper secondary and tertiary structures. So to get it folded in the proper conformation, it's been estimated that that's a 1 in 10 to the 74th chance, okay? And then we need to get all 150 amino acids in the proper order, right? You could have 150 amino acids, and if you actually made it through all these probabilities, you would have them all in the L form, all peptide bonded together, but you need to get them in the proper order. I mean, that probably should come before the functional shape, but that's a 1 in 10 to the 195th chance, right? If every time you add a new amino acid, you've got another 20 to choose from, that's 150, and for every one of the 150 proteins, you've got a 1 in 20 chance of picking the right one, right? If you do the math, that's a lot of options to get them all in the right order, okay? To give you a comparison of this, well, so to total it all up, so in the prebiotic soup, and just before life was formed, to just get a functional amino acid by chance, if you add these all up, and we're not even talking about the proper order, to get any protein together in an actual form that proteins take, right? Regardless of the actual sequence. Let's not even say it has to be a particular sequence, just say, let's just get a protein in a functional shape. 10 to the 164. 10 to the 164 power. For comparison, there are only 10 to the 65 atoms in our galaxy. Okay? So imagine picking through every atom in the galaxy. You're still not even approaching the high unlikelihood of getting a functional protein to just assemble by chance, right? There's only 10 to the 80 protons, neutrons, and electrons in the entire universe, okay? Now, if those aren't staggering enough, there's only been 10 to the 16th seconds that have passed since the Big Bang. So even if you could choose this, there haven't been, you're doing 1 per second. If you did 1 per second, you still are not even coming close to the 10 to the 164 power, okay? So it can't just be chance, right? This has actually been ruled out by most naturalists, right? Nobody believes that you can just get an amino acid polymerizing together by chance. The numbers are just overwhelming, right? So if you can't do it by chance, well, and then, okay, RNA polymerase, you have to have RNA polymerase in a cell, right? If you're going to make mRNAs and make any proteins, this is 3,000 amino acids, right? So think about, this is just for 150 amino acid protein. Think about how big these numbers are going to be if you're talking about a 3,000 amino acid protein. The numbers just are staggering. The simplest cell that we can think of possibly making, if you just like took Mycoplasma genitalium and you just kind of started throwing out proteins that you think probably don't have to be there to just basically just survive, we think that probably the simplest cell you could probably narrow down to maybe 250 proteins. So think about the staggering things for just one 150 amino acid protein. Most proteins are going to be more, thousands of them, and you have to have at least 250 of these, right? The numbers just become ridiculous, right? So getting proteins to just polymerize together to have the necessity of life is just, it's just impossible in terms of statistics, right? There's just not enough milliseconds over time to have been able to choose all of these, right? So, yeah.
So how come a lot of scientists still choose to believe that there is no...
How come people still choose to believe that there was no design? I mean, that's a philosophical question, right? I mean, this is the data, right?
Yeah.
But what do you conclude from that? I think it depends a lot upon your preconceived philosophy, right? If you're already committed that there is no God and that things work about by naturalistic processes, then you're going to have to come up with another way. You know, well, this isn't going to work, but I'm committed to my philosophy, so I'll find another way. I mean, to be honest, in the same way, I'm committed to my theism for a lot of other reasons besides this, too. So this reaffirms my theism and say, yeah, I'm pretty convinced that there must have been some active intelligence to do this, because chance can't get it for you. But I think actually few people are really convinced by data necessarily, right? How you're interpreting that data based on your philosophy that you've got lots of other reasons for believing comes really into play. So you start talking about people's psychology and their background and their reasons for interpreting data in certain ways. So, I mean, that's the art and that's the supernatural power of evangelism, right? You don't, hardly anybody, I don't know of anybody who's actually come to Christ or come to believe in God just because somebody showed them a bunch of arguments and statistics, right? People just don't function that way. So, I mean, this could be a powerful tool to break down people's barriers and make them open to maybe considering it. But deep down, why people don't believe, it's more complex than that. So, if chance alone is not sufficient to bring about functional proteins, then there's got to be something else. If you're committed naturalist and you're not open to the idea that somebody designed this, then it must have to do something with chemical necessity. If chance alone can't get it to you, right, you have these huge improbable numbers. And I think it was Richard Dawkins that actually wrote a book called Climbing Mount Improbable, where actually he plays out all these improbable nature of life spontaneously arising and said, how do you climb that mountain of improbability? Now, I'm not going to talk about Dawkins, but part of that means that there's some necessity, right? There's some physics and chemistry that impose order on nature, right? So it's not just completely random chance to get functional things, there's laws of physics and chemistry that say it has to be this way. This is Dean Kenyon. He was a chemistry professor, San Francisco State, I believe is where he was. And in 1969, he wrote a book called Biochemical Predestination. And he was really wrestling with this fact of like, okay, chance alone is not capable of assembling functional proteins together. So how did the first functional proteins actually arise? And so he said in biochemical predestination that amino acids have certain chemical properties, there's certain chemical and physical properties to amino acids that make them polymerize spontaneously, self-organize themselves into functional proteins, right? So he wrote an entire book kind of talking about the laws of physics and chemistry that he thought were sufficient to say, well, it's not just chance, physics and chemistry dictate that functional proteins will arise. The problem with that is the protein sequence is what I'm calling contingent, right? The only way that amino acid sequences, or the only way that nucleic acid sequences can contain information is if they could be otherwise, right? If I'm writing in a medium that dictates that an A always has to follow a T, right? Then I'm just going to say TA, 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 TA. And I'm going to have some information, but it's going to be the lowest level, the lowest content of Shannon information that you can think of, right? TA, 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 huge long string, can just be reduced down to TA 100 times, right? That's not much information there. For things to actually hold information, you can't have to have a chemical rule that says a T always has to follow an A. Anything could follow a T, right? That's what's allowing you to put information in it. So protein sequences cannot be based on physical chemical laws, or you're not going to get anything. Right? The other thing is, how do proteins copy themselves? Proteins can't copy themselves. Proteins have to have DNA and have to have an RNA, and proteins copy the DNA and RNA and turn it into other proteins, right? So I've got this functional thing where, say in fact you did, by chance or by necessity, get a functional protein together, how is that ever going to copy itself, right? Getting a second amino acid is going to have to do all those same things, right? There's no functional way for them to copy themselves without there being functional information there already. Functional information in the DNA that tells you how to copy it into the amino acid. So that's a problem. You have to have DNA. Now, how do you get DNA? You run into all the same problems now of how do you get a functional piece of DNA? All those statistical things. Now, it's a little bit easier in terms of probabilities because there's not as many options in DNA. Right? There's 20 amino acids. So for every position in a protein, there's 20 different amino acids that could be there. In DNA, there's only 4 different nucleotides that can be any spot. But still, the chances of something spontaneously arising are still highly improbable. And so DNA and proteins seem to be intricately related. Right? You can't have one without the other. This actually convinced Dean Kenyon that his worldview was probably not correct. And Dean Kenyon actually, years later, totally recanted everything he said in his book. He said, that just can't be. That doesn't explain information. And Kenyon, I think he's still alive. He's quite an elderly man if he is still alive. I haven't checked. But he actually, through other circumstances, actually became a believer And so he was an avowed atheist. He became a believer. And actually, I think it was San Francisco State. Now, I'm saying this on the internet, now I'm doubting it. The institution that he was at actually took him out of Gen Bio classes and chemistry classes because he started presenting the evidence for an intelligent design being necessary. And they said, you know what, we don't want that being said to our students. So he was actually, his teaching positions were taken away. And they didn't fire him, but he couldn't teach his classes anymore. So he's an interesting character. He's written a couple of books since, and he's been one of the really early advocates for an intelligent design model. This is Alexander Oparin. Oparin kind of models the combination of necessity and chance together. Okay, so chance alone isn't sufficient. Physics and chemistry alone can't account for information, so neither of those are complete. Oparin said, well, maybe chance and necessity together can explain. Some things arising by chance, other things arising by necessity. So the kind of prebiotic natural selection is what this is called. So you've got, before cells, you've got prebiotic molecules coming together. And so Oparin said, well, maybe by necessity, certain atoms have to come together and form amino acids. So chemistry and physics alone, just by chance, can account for that. So that's necessity. Then by chance, these molecules form proteins. Now we've already kind of poked holes in the fact that, okay, even if you have 20 amino acids, can chance really, is it statistically significant to say that chance could actually get you a functional protein? I don't think that one holds up. But the idea is, well, necessity dictates some things come together. By chance, the molecules form proteins. By necessity, and by necessity he means natural selection, those proteins, the only ones that were functional are the ones that survived. So this is his trying to say, well, you can get random proteins coming together, but only the functional ones stick around. The problem still holds up is proteins by themselves can't replicate themselves. And for natural selection to be occurring, you have to have things reproducing. That's what natural selection says is, right? The next generation of offspring have to be selected against. Well, if the random protein, well, if the protein that came about by necessity and chance can't replicate itself, natural selection doesn't help you, right? So I think number 3 is faulty as well. By chance, these molecules maybe came together in these primitive protocell membranes. So you got some of these amino acids and DNA and nucleotides together in a membrane, and then that started being functional as well, and natural selection continued to work. Now, I think that's very problematic too, because I would want to say that the cell is actually irreducibly complex. Like getting some proteins and some nucleic acid in a membrane is not going to give you a functional cell. So this is kind of the only options left to you, though, if you're a naturalist, right? Either chance, either necessity, or the two of them together. There's nothing else that you can really postulate that's a naturalistic view, right? That's as a naturalistic view, you're saying physics and chemistry are the only things that are acting in the universe, right? If you add anything additional to that, like a Gaia principle or self-organization tendencies or things like that, you're starting to talk about kind of quasi-spiritual things, like something other than the physics and chemistry is doing something. If all you're saying is physics and chemistry, all you got is chance or necessity. And if those are inadequate, then you're going to have to go to something else. As theists, we believe in a personal God who did this, right? But there are other New Age, spiritual kind of ideas floating around there as well. I don't think any of the paganism of old is really dead. It just shows up in sophisticated ways with kind of modern language associated with it. But there's still a lot of mystical ideas out there too about how life arose. So, yeah, Rachel.
Mm-hmm. How are they saying that those atoms do they have?
Yeah, I don't know actually enough cosmology and astrophysics to give you an adequate explanation for why atoms came together at the Big Bang. Yeah, I'm just inadequate to answer that question. I'm sure there's theories about it. I just don't know.
I'm just curious if they actually use it.
Yeah.
Because I've always kind of said that.
Yeah, I'd send you to John Bloom. He's probably thought about this more than I have. So yeah, yeah. All right. My view is that the inference to the best explanation for this is that codes like amino acid codes and nucleic acid codes have coders. Information comes from intelligences. So I think the best analogy is when you find a code in biological things, you don't say that physics and chemistry did that. You say a coder did that. If you find a code in a computer, you naturally think somebody coded that, right? A group of people, an individual. Programs, like the programs that go on the cell, right? The cellular program to divide, programs have programmers, right? So programs are made up of multiple individual codes. Designed systems, like organisms, like ecosystems, designed systems have designers. And this is, the question comes, is this science or religion, right? Because as soon as you start saying things that are outside of physics and chemistry, people will say, you're now bringing your religion into it, and religion has no place in science. And I don't think that's correct. Because what we're after is, what is the truth, right? And if the truth has religious implications, then so be it, right? I don't think we should limit our worldview to just things that physics and chemistry can explain, right? If we find some true things about the universe and they have religious implications, then we have to follow them there, right? If you're denying them, then you're just putting your head in the sand, right? And ignoring things. So what I'm after is not science or religion, I'm after truth. And if that leads me to a religious view, then yes, I'll follow it there. So this is actually one of the ways that people come about truth in both science and religion. So this is why I've titled this slide, Inference to the Best Explanation. If you've got a phenomena and you want to explain it, then you may have to infer to what the most likely story, what's the best story to fit it, right? I might not actually have scientific evidence for it, but what's the best story to fit it? That's a good way to come about trying to decide what is true, right? So based on the empirical evidence, I say that inference to the best explanation is that things must have been designed. Now, this gets used in all kinds of sciences and all kinds of other disciplines as well. Inference to the best explanation is often used in historical sciences, right? If you're trying to think about, what was the motivation for somebody in history doing things, you can't go ask them, nor can you do a test to see if that same person would make the same decision, right? I mean, that person's dead, right? So just because you're making an inference to the best explanation doesn't mean you're a quack, right? Oftentimes, as soon as you label the term or label the thing religion, then obviously it means it's completely devoid of any rational thinking. That's what most people say when they say you're bringing your religion into it. Well, if my religion makes truth claims and is doing truth seeking based on some, methods that are used in lots of other disciplines, then I'd say that's a good thing to do. That's a good way to test your religion, right? So historical sciences, you can make predictions based on what you think. So any attempt to call inference to the best explanation for an intelligent designer, if you say that that's not a scientific thing to do, then you're actually going to rule out a lot of other sciences that actually use inference to the best explanation as well. So attempts or disciplines like archaeology, if you find some bones and you're trying to recreate an old historic ecosystem or some type of physiology of an animal, you're making inferences to the best explanation because you can't do a test, you can't recreate that anyway. So archaeology does this all the time. Paleontology does this time. Cryptography, if you're trying to break codes, you're using inferences to the best explanation and implications of what the coder did. So that cryptography is a science. SETI, the search for extraterrestrial intelligence, that's using the exact same type of methodologies that intelligent design is looking for, right? SETI is looking for, are there any intelligent codes being sent to us by extraterrestrials? I think the government slashed this pretty bad, but I think the government actually still does fund SETI research to see if there's any extraterrestrial life. any evidence that extraterrestrial intelligences are sending us codes, right? And most people want to say that that's science, right? But it's using the exact same methods that we're using by just looking inside the cell, right? So whether the information is coming from distant planets or coming from the inside of a cell, we're still looking for codes and coders, right? So other sciences too, I won't go into all of them, but they use these type of inference to the best explanation, looking for coders, looking for intelligent explanations for phenomena. So, all right, I blew through this. You guys want to feedback with me on this at all? I just started getting excited, so I quit asking you questions. So, yeah.
What are your thoughts on directed panspermia?
Directed panspermia is the idea that extraterrestrials actually planted primitive cells on the Earth, and that those cells, since they're functional and can reproduce, then natural selection on our Earth could create the diversity of organisms that we see. Fine, valid hypothesis. Is there any evidence for it? I don't think there's any evidence for it, and I don't think it answers any of the fundamental questions of origin of life either. Because so what if life got put on this planet as a single cell? If we're thinking about the origin of life and the origin of cells, how did those extraterrestrial cells come about, right? It seems to just punt the question to another planet. But all the same questions still arise. Right? How does functional information come about? How do organisms design themselves? How did the first cell arise in that extraterrestrial? Unless there's some other wacky dimension where the rules of physics and chemistry are different, I don't know how it really explains anything. So, yeah. I give it that it at least acknowledges that it's really hard to think about how anything could spontaneously have arisen on our planet. It really does look the inadequacy of chance directly in the face and say, yeah, you can't come up with functional cells and functional proteins just by physics and chemistry here. But I don't think it answers the question. It just punts it someplace else. So, yeah.
Just curious, you know if that's a very widespread theory or if that's
No, it's not very widespread. No. I mean, it was put about by some famous people, like Francis Crick, for one, was a big advocate of the idea of directed panspermia. Yeah, does Dawkins talk about it too? I think Dawkins mentions it. I'm not sure where he comes down on the issue. I think he's pretty convinced that maybe life could have arose here on Earth without being planted, but yeah. Sure.
So when we make the inference to the best explanation, which I totally understand, and I think that we all do it, we're saying that everything we don't understand about some of these issues, it could be the origin of life, or it could be how evolution got us from monkeys to humans if you're a naturalist. Why don't we decide that those, and I know what you're going to think I'm going to say, but those gaps are, why do we think that those are God there? When none of the other gaps we used to think were there, for instance, the structure of DNA or how does a cell reproduce, things that in the past we didn't know, but now we know. Why are those things that we think are now God, not just things that we haven't yet?
So the question is, why do we infer God for things we can't explain right now, when in the past we've had things that we can't explain, but now we can?
We've had several naturalistic explanations for almost everything we understand.
Well, I would disagree with that, but yeah, there are certain things that we didn't know how they came about, and perhaps, and I don't know enough history to give you examples, but maybe there were issues where we didn't know how this arose, therefore God must have done it because we don't understand it. I think this is different because this is not an argument from ignorance, and I think the God of the gaps argument is an argument from ignorance. It says, I can't imagine a way for this to happen naturalistically, so therefore God must have done it. I don't think that's what I've done here. What I've done here is say, I think I do know how these things come about. I think I know that codes come from coders. I think that programs come from programmers. So I'm not saying I don't know how it happens. It's saying I know of actually really good examples of how things come about that are beyond physics and chemistry. So the best inference is to say, I think an intelligence did it, just like I think an intelligence writes codes in computers. So I would say that it's an inference to the best explanation, not an argument from ignorance. So I don't know if I made those two distinct enough. But I'm not saying it's just because I can't imagine naturalistic explanations. It's saying naturalistic explanations are inadequate. A better explanation is an intelligence. So I'm going to go with the better explanation. Maybe we could feedback on this at some other time. But what I'm trying to show is that this is not an argument from ignorance. It's saying, I don't know how this can come about, so somebody must have done it. No, actually, I know a lot of examples where things like this do come about by intelligences. So therefore, I think an intelligence is the better explanation. Maybe that sounds the same to you, but we could talk about it. Rachel, I'd love to take your question, but we got to move on. So, or else we'll be 3 lectures behind. So we're going to talk about Mendelian genetics now. So I was going to say reset your brains. Don't reset your brains, because I think this is continuing the same theme. But we're going to talk about Gregor now. Talk about Mendel. We talked about Mendel before, Augustinian monk, and he did his pea plant experiments in 1856 to 1868. And it was through these pea plant experiments that he created what he called his laws of heredity. Okay, this is in contrast to what Darwin was proposing right around the same time, that idea of gemmules passing on that you guys talked about earlier. That we talked about earlier. All right, so why did he pick pea plants? Next Friday, we're going to plant some pea plants in honor of Mendel in the organic garden. But he picked this pea plant. And I always goof up the Latin name here, Pisium sativum. This is just a common pea. There's a lot of varieties of this, but like the sugar snap pea, the snow pea, a bunch of other peas, they're all the same species, just some different varieties of them. Okay. Why pick the pea plant? It's actually a really great model organism. It's really easy to grow. It fixes nitrogen in its soil, so it can grow in pretty nitrogen depleted soil. You don't have to have great growing conditions to grow peas. So if you're starting off gardening, one of the greatest things to do is grow peas, because they just grow wherever and you get lots of them, and it's pretty satisfying and encourages you to keep gardening, right? They have a lot of offspring. So for every pod, so here's the little pea pods. So here's the flower. There's different varieties too in multiple phenotypes, and they're pretty distinguishable as well, which makes it a nice thing to do genetics with. The typical wild type has purple flowers, but there's also white flower varieties and pink flower varieties, and they're clearly distinguishable, right? It's not a subtle difference, it's pretty stark contrast. They have pea pods, and in each one of those pods are multiple peas, and each of those were the product of a single fertilization event between an ovum and a pollen, right? So you can get lots of offspring. They make just hundreds of pods in one season, and you can just get a lot of data. They're easy to grow, okay? They're easy to cross-fertilize, which makes them really, they're easy to control the cross-fertilizations, and it's easy to self-fertilize them as well. And one of the issues with this is actually the timing of when things come out. So you can actually, so here's some pollen, botany, anthers, is that correct? The pollen is in the anthers?
Yes.
Yeah, and the ovules are going to be in the flower part of it. Those are two different organs on the same plant, and they come out at slightly different times. So if you don't want a plant to self-fertilize, you can go clip off all of the stamens, remove all of the pollen, and basically, like you've been searching for virgin female flies this week, you can basically make virgin female pea plants by just clipping off all of the anthers, and so it doesn't self-fertilize. Then you can come in with a little paintbrush and grab pollen from a different plant and fertilize the female plant that you want, so you can control the breeding very well. So it's actually a really nice, nice plant to work with. So here's an example of the kind of experiment that Mendel would do. He would look for what he called character. A character is a physical feature of the plant. So flower color. Flower color is one of the characters, okay? And then he would look for what he called particular traits. And that would be the actual particular form of that character. So if the character is flower color, the trait would be white flowers versus purple flowers. right? There's several different really distinguishable phenotypes in the pea plants, and then this will show one of the single peas can either be smooth or it could be wrinkled. Okay, very clear traits, easy to distinguish. What he would do, well, actually, let me fill this in. I just totally lapse into modern language, so I'm just going to show you the modern language so that if I get things confused, then you'll know what I'm talking about. When I say a character, we're going to think of a character being coded for as a gene, right? There's a gene located on the chromosome that codes for flower color, okay? And the different traits that we see, white or purple flowers, smooth or wrinkled seeds, are because we have different alleles, different versions of that gene sequence, okay? So this will map you onto what the terms that Mendel used, but I kind of just lapse into this, so I just am clear to begin with. So, here's one of the things he would do. He would take a pure breeding smooth pea plant. If you take this plant and continue to self-fertilize it, or you take all smooth plants and fertilize them together, and if what they always come out with is more smooth seeds, then you have a pure breeding strain. So every plant in that patch of garden that he had, if you crossed all of those together, the only offspring in the next generation would all have smooth colored seed or smooth shaped seeds, right? So pure breeding. You can either have a pure breeding individual if it's self-fertile, or you'd have to have a pure breeding group or strain. And then he would take a plant that was true breeding for wrinkled seeds. So spontaneously, just as people have been growing peas, they notice, oh, some of them are wrinkled. Well, if you grew up that plant and you grew up another plant from that had a wrinkled seed, and if you continue to cross those together, they always come with wrinkled seeds. So to do a really good genetic cross, you have to have pure breeding strains. Actually, that's what I've given you in the flies that you're working with. You've got pure breeding wild types, and you've got pure breeding black-bodied vestigial wings. So no matter how many times you let those black-body vestigial wings breed together, they're always just going to come out with that same phenotype. So Mendel took these two plants and he cross-fertilized them, right? So he took pollen from one and fertilized ovules of the other in a controlled way. And what he saw was that the traits, and by traits we mean the alleles, either wrinkled seeds or smooth seeds, did not seem to blend, right? Darwin's theory was already out there. Darwin says depends on the gemmules you inherit. So if you took the gemmules from a smooth and the gemmules from a wrinkled, you should have a moderately wrinkled offspring, right? Because the gemmule percentage would be 50-50. You come together, you should have a almost right in between phenotype, right? A blending phenotype. But Mendel saw that there was no blending of these traits. The alleles were either dominant or they were recessive. So here, you took the pollen from the wrinkled, the ovule from the smooth, you crossed them together, and what you came out with in the next generation was all of them were smooth. The wrinkle phenotype wasn't there, wasn't subtly wrinkled, it wasn't blended, they were all there. And he'd say, okay, well that's a dominant trait then. That trait completely masks over the wrinkled, okay? Now what he would do is he'd take that plant and he would cross it to all kinds of different things, just testing to see what's going to happen. One of the things he would do is fertilize it to itself, right? What would happen then? If you fertilize it to itself, the results, and this is the second generation now, so this is F1 or the filial generation 1. This is the F2, the filial generation 2, that's from the self-fertilization. And what he saw is the wrinkled phenotype, the wrinkled trait came back. And it came back just as robustly wrinkled as when it originally was in that parent generation. There again was no blending. It's either there or it's not there, just as wrinkled as it was before. So he called that trait a recessive trait. It's in the background, it gets covered over, but when it comes back, it's just as strong as it was before. And he noticed that this was in a characteristic 3-to-1 ratio. So in that F2, for every 3 smooth, every 3 dominant he saw, he would see 1 wrinkled. Now this is careful observation, controlled breeding, good methodology here. The real clever thing that Mendel came up with, though, is an explanation for why this is true. And what he did was kind of the classic thing that you would love to do in biology, is like map it onto mathematical rules. And that's what he did. He mapped this onto mathematical rules, and he called it his particulate theory. We just call this now Mendelian genetics, but he called it his particulate theory. He said for every character in an organism, the plant has 2 discrete particles that are going to determine what trait that character shows up as. Right? So for the character of of shape of seed, there are 2 things that are going to determine whether it's wrinkled or whether it's smooth. Okay? Every plant has 2 discrete particles. And each gamete inherits one of those 2 particles. So a smooth is going to give smooth character to either its ovule or its pollen, right? So we can represent this. So a big S would be the particle that codes for, or that determines smooth character. And little s is the particle that is for wrinkled, right? So this would be representative of the original cross that he did. He took a smooth, pure breeding smooth strain, so it must have each had the same particle, same smooth particle. And he crossed it with a pure breeding wrinkled, same wrinkled particle, okay, small s. And the F1 generation must have had one of each, okay? Inherited 1 from an ovule here, inherited 1 from a pollen there, and so its genetic contribution is wrinkled and smooth. But this is going to look smooth because the dominant character is smooth, right? And it's going to mask the recessive. These we call homozygous because they have 2 of the same. These guys we would call heterozygous. They've got 1 of each, okay? So that second round of experiments he did is he took one of these heterozygous guys that had 1 of each particle, crossed it with either itself or somebody else that had the exact same background. And then what you get out of it is 3 different ways you can combine them, right? I could have a big S going with a big S, that would give me a homozygous. Small s going with a big S, that's going to be a heterozygote, or the 2 small s's going together. And this is what's going to determine your character or your phenotype. These 2 are going to look smooth. This guy is going to look wrinkled. And this gives you your characteristic 3-to-1. This explains why you can do 3-to-1. For every 3 wrinkle, or every 3 smooth, you see 1 wrinkled. Because that big S could go with, big S could go with big S, that's 1. That small s could go with a big S, that's 1 of those. This big S could go with that small s, so you can get 2 ways that you can get the heterozygote. Only 1 way you can get that. So if that's 1 individual, that's 2 individuals, that's 3 to 1. So his particulate theory gave you an explanation for why you always saw 3-to-1. This was really Darwin's critical insight. And he just did this by playing with the numbers and saying like, well, let's just come up with a mechanism that would give me 3-to-1. This happens to be correct. We know it's correct because we know what happens on chromosomes. And in the next lecture, we'll actually talk about why chromosomal theory explains why Mendel's rules are true. So, all right.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.